Well, what if you could edit your DNA just like how you edit a text document, removing unwanted parts and adding new ones? Sounds like science fiction, doesn't it? But this is precisely what the revolutionary technology known as CRISPR allows us to do. You see, DNA is the blueprint of life. Every cell in our body contains a copy of our DNA, which is a complex code that guides the production of proteins, thereby defining our traits. From the color of our eyes to our susceptibility to certain diseases, it's all scripted in our DNA. Enter CRISPR, a technology that allows us to make precise edits in this DNA code. The origins of CRISPR are quite fascinating. It was discovered in bacteria, serving as a sort of immune system, a defense mechanism against viruses. Bacteria would capture snippets of DNA from invading viruses and use them to recognize and combat the same virus if it attacks again. The discovery of CRISPR opened up a Pandora's box of possibilities, changing the landscape of genetic engineering forever. CRISPR, a technology that has been hailed as the biggest game changer in genetic engineering, was discovered in the 1980s, but only understood in the 2000s. What began as a mysterious sequence of repeating DNA found in a simple bacterium took the scientific community by storm when its potential was realized. It wasn't until the 2000s that researchers understood these sequences were part of a sophisticated bacterial defense system. As the new millennium dawned, scientists found that this system could be harnessed as a powerful tool for genome editing. The first successful experiment using CRISPR to edit a genome was carried out in 2012. This marked a significant milestone in the journey of CRISPR, turning the tide of genetic engineering. Today, less than a decade later, CRISPR has evolved from a fascinating biological oddity to a tool with the power to revolutionize medicine. This is the game changer, the tool that is rewriting the rules of genetic engineering and reshaping our world. From treating genetic diseases to improving crop yield, CRISPR is already making waves in various fields. Imagine a world where genetic disorders like sickle cell anemia could be cured before birth. This isn't science fiction, it's the power of CRISPR and Cas9. Scientists have used this technology to correct the mutation that causes this debilitating disease, offering hope to millions worldwide. But this isn't just about medicine. In agriculture, CRISPR is being used to create crops that are more resilient, productive, and nutritious. Picture a future where food scarcity and malnutrition are things of the past. Livestock breeding too is being revolutionized. Scientists are now able to edit the genes of livestock to make them healthier and more productive. The applications of CRISPR are vast and varied, and we are just scratching the surface. From our health to our food, this groundbreaking technology is shaping our world in ways we can't even imagine yet. The future of medicine lies in our ability to manipulate the building blocks of life itself. Imagine a world where we can eradicate hereditary diseases that have tormented generations. This is not a far-off fantasy, but a potential reality made possible by CRISPR technology. We could enhance human capabilities, increasing our longevity, boosting our intelligence, or even engineering resistance to diseases. The possibilities are as vast as they are thrilling. Yet, with great power comes great responsibility. The ability to edit life's blueprint brings forth a multitude of ethical considerations. We must tread carefully, ensuring that this technology is used for the benefit of all and not the few. We must also grapple with the potential consequences of resurrecting extinct species, a prospect that is equally fascinating and daunting. As we stand on the brink of a new era, CRISPR holds the promise of a future where we are no longer bound by our genetic destiny. And if you're as excited as we are about the future of science, don't forget to subscribe for more fascinating content.